Let us start now in the very important pre-fertilization event of a flowering plant, that is about stamen, Microsporangia and pollen grain. What are you going to do in this chapter? We're going to discuss in detail about the structure of the stamen, structure of the Microsporangium, and we'll also talk about the structure of the pollen grains. So there won't be anything left behind over here. We're going to discuss each and every part of this particular topic in detail. Okay now? So let us see what exactly it is. Stamen, Microsporangium and I went to the previous slide. Yeah. Okay. Now, as we told you, Androidium is the male reproductive part of the plant, right? And what it is made up of? It is made up of two important parts. That is the filament and the anther, which is present on the top of it. The filament is also called as stalk, and the anther is basically bilobed in nature. So Androidium is the male reproductive role of the flower and it contains the stamens. Okay. Each stamen is made up of three parts: filament, anther and connective. Though connective is not visible in this particular photograph, but there is a part which is called as connective. Let me show you this connective. If I can draw this in a proper way, I think I can show this to you. Okay. Let me go to the slide and let me zoom the picture for you. Okay now. Okay. Now, what is it? This is the male reproductive part of the plant. Okay. Now, what is present in the male reproductive part? First of all, what it is called as a male reproductive part of the flower is called as Androidium. Okay. And in the Androidium, we have the filament. So this part is the filament. This part is the filament. What is the filament over here? Filament is this long stalk which is actually in turn connected to the thalamus part. Here you will be the thalamus. Okay. If I wanted to draw it, properly, see, if this is the thalamus, can you see the thalamus? Thalamus, and this is the particular pedicel of the flower. Okay. So this is the pedicel and this is the thalamus. Okay. On the thalamus, above the thalamus is a landing surface type, flat type. On the thalamus, there will be some stalks which is going to come up, and these stalks are nothing but the filaments. Okay, so you will find out these type of stalks present on this, and on the top of it, you will find out these bilobed structure. Those are called as anthers. So what is a stalk? Stalk is the particular long tube-like structure which is basically holding the particular whole anther part to the thalamus part. So here there will be the thalamus part, and then on the top of it there is anther. What is anther? Anther is having two lobes. You know lobes? Lobes means like two halves. Two circles are connected together. In between the two lobes there is a typical tissue. Those tissues are called as connective. As the name says, it is for connection, right? Connective means to make connections, isn't it? So there in between the two anthers there will be a tissue part that is called as connective. So what is there we have? We have this in the pollen grain structure, stamen, Microsporangium and pollen grain. If I talk about it, Androidium is the male reproductive part of the role of the flower which consists of stamen, and each stamen is made up of three parts, that is the filament, anther and the connective. What are the three parts over there? The three parts are filament, anther and the connective. Okay, so filament is a long slender like part like this. Anther is the bilobed structure. In between the anther part there will be a typical tissue that is called as connective. Clear? Let us go to the next slide, what it is called as. And okay, one more thing I have forgotten to tell you over here, that this is the transverse section of the stamen, TS of the stamen, that is transverse section of the stamen. If I cut open the stamen, I mean the pollen grain part, the anther part of it, we will be seeing that in that there are four thecas or four lobes. It has two. Anther is actually bilobed in nature. It has two lobes, and you will find out these type of structure. Okay, in this particular part there are four parts. You can actually make it four section over there. I think it is not visible. Let me clear everything and I'll show you. Okay, I'll change the color of the part. Okay now. So this is the part. Okay, can you locate this part now? Yeah. In this particular part, you will find out four parts. There will be four circles type. Okay, like this type. And this is actually the part where the pollen grains are stored. Okay, na? So if I wanted to dissect a flower, if I want to dissect an anther, if I just want to cut the anther like this, then I'm going to see these type of particular structure, which is actually a transverse section of the anther. Inside this we'll find 4 lobes. I'll talk about it in tetrad formation. I'll tell you in the next slide. Inside that there is an anther formation. Now as it is in this picture, you may ask that, ma'am, you didn't explain only about this. So what exactly it is? It is the transverse section of the flower. Okay. Next, let us see what is the next one. Okay. Filament is the part which is holding the anther. As I told you, filament is a long slender part which is holding the anther. Filament and anther lobes are joined by the connective tissue. The filament and the anther lobes and the anther intermediately also are joined by a tissue that is called as connective tissue. The lower end of the filament is attached to the thalamus. I just now I told you, right? The lower end of the filament is connected to the thalamus. So thalamus, where the flower, all the parts of the flower basically start from the thalamus. So lower end is connected to the thalamus, and then upper end what we get this 2 bilobe structure that is called as anther. Okay. That is about the structure. You should remember all these points. Okay. Now each lobe of anther has 2 pollen sac, or called as Microsporangia. What is Microsporangia then? Microsporangia is nothing but the pollen sac. Did you understand? Microsporangia is nothing but the pollen sac. Such type of anther is called as dithecous. What are they called as? Dithecous. Theca meaning that particular anther. Each lobe is called one theca. Then ditheca means 2. As we see in case of pea plants. If you see the pea plants, it has 2 thecas, and that is why it is called as dithecous. So each lobe of anther has 2 pollen sac or microsporangia. That means pollen sac and microsporangia are the same thing. You should not have any confusion. What is microsporangia? This is a new term I am learning. I never heard about microsporangia. Microsporangia is nothing but the pollen sac. What is the pollen sac? Carries pollen grains, obviously, right? So a sac-like structure which carries the pollen is called as pollen sac, and that is also called as microsporangia. Okay. And this type of anthers, if it has 2 lobes, then what it is called as? It is called as dithecous. In certain families like Malaceae, the anther may be monothecous. Monothecous means it will have only 1 theca. It will have only one particular theca. It won't be having 2 thecas. In case of pea plant, you will find 2 thecas or 2 lobes connected together. In the family of Malaceae, you will not find 2 theca, but rather you will find 1 theca, and that is called as monothecous plant. Example is hibiscus. So what you have to remember from here? You have to remember 2 things. Plant anthers can be of 2 types. That is, one is monothecous type, one is dithecous type. Monothecous means single theca or single anther part is there. Example, hibiscus. Dithecous, which is pea plant, where it has 2 thecas which is connected together. Okay. Is it clear to you? Did you understand this slide? That each lobe's anther has 2 parts, monothecous and dithecous. Now, the bilobe nature of an anther. Bilobed. Bi means 2. Lobe means part. Okay. Bilobed nature of anther is very distinct. It is very distinct, and it can be seen in the transverse section of an anther. If you do a transverse section of anther, you can clearly see the 2 lobes present on the anther. This is the one which is showing 2 lobes. So if I actually transverse section, if you do this, you can basically make out there are 2 lobes. Okay. 2 lobes are present on the anther. Okay. The anther is 4-sided tetragonal structure. What is the anther? Anther is 4-sided tetragonal structure consisting of 4 microsporangia located at the corners, 2 in each lobe. This sentence tells you the complete meaning of this particular picture, if you understand this properly. The anther, what is it? Has 4 tetragonal structure, or it is something like this. Okay, the structure will be like this. Tetragonal structure consisting of 4 microsporangia. So there are 4 microsporangias which is located like this. Got it? How many microsporangias are there? 4 microsporangias are present. What is this? This is microsporangia. Okay, now. What is microsporangia? Just now I told microsporangia are nothing but the pollen sacs. Clear? So, the anther has 4-sided or tetragonal structure consisting of 4 microsporangia located at the corner, 2 at each lobe. 2 at each lobe. You can see 1 and 2 over here, 1 and 2 over here. 2 at each lobe. That's what exactly the same thing, whatever is in the picture. Clear? So, a bilobe nature is very common, and when we see in the transverse section, we see that there are 4 structure, tetragonal structure which is present, and they are called as 4 microsporangia. Okay. Now, these microsporangias will be developing further to form the pollen sac, or pollen sac and microsporangia is the same. Microsporangia itself will develop to the pollen sac. Now, this is the one. Can you recall? I just now drawn. There are 4 tetragonal structure. Okay, now. Now, what happens in this? They extended longitudinally all through the length of an anther, and they are packed with pollen grains. Okay. So, if I wanted to show you like this. Okay. This is the previous one. Not this. Not this. This. Yeah, this one. Yeah. Now, if I wanted to show you this particular picture which is there on the screen, see, can you see the pollen sacs or microsporangia over here in this particular photo? There are 4 microsporangias. Yeah, let's get this visible now. Okay, it's written 4 pollen sacs. Now, here what happens, you know, when the flower is growing, at that time when the microsporangia need to develop, all these 4 microsporangia will start developing. Nearby, there will be so many layers. You can see these layers. Can you look at these layers over here? Can you look at these layers? Yeah, there are 3-4 layers. I'll tell about the layers. What are the layers and what are their functions? Everything I'll explain you over there. But can you see this? These are the part where they are completely filled with microspore mother cells. These microspore mother cells, or which is also called as MMC. Microspore mother cells is shortened as MMC. They are containing the pollen grains. At one particular time, what will happen? The whole anther will be filled with pollen grains. So, these 4 particular part or microsporangias are actually forming pollen grains, and these pollen grains will be filling this particular part completely. Okay, so they will be making so many pollen grains. So, they extend longitudinally. They will extend longitudinally all through the length, and they will be packed with pollen grains. What will be packed? The microsporangias. Okay, so microsporangias will be packed with the pollen grains, and they will be extend longitudinally over there. The bilobe nature of the anther with 4 pollen sacs can be seen when a transverse section of anther is observed under microscope. If you cut an anther, if you just do a dissection and observe under microscope, put it in on a slide, then you can see this 4 structure very clearly. The structure of microsporangia is visible clearly if you do a dissection of the microsporangium, and then you can check it out. Okay. Did you understand this? Now, as I told you, there are layers, right? There are different teguments. Teguments means the covering part. Okay. So, now when we talk about the complete structure of microsporangia, if I do a transverse section of typical microsporangia, it will appear circular as an outline. Okay. It will appear as circular outline. I think almost, yeah. Can you see now? Okay. Can you see this? There is circular outline, and if I wanted to take that particular part and enlarge it, then it will be looking like this. Okay. One thing I want to tell you that you have to practice these diagrams. Okay. You have to practice the diagram of a typical flower, then the structure of microsporangium, and this is very important diagram. In exam, they ask this. So, please practice this diagram. The same diagram is also there in your NCERT textbook. You can take it from there also, no problem at all. But you have to practice, because in exam, if it comes for 5 marks or more, then at that time you have to actually draw the diagram and explain.